when the line is not fast enough, there is a delay. So I will now click. When I say top, I click. And you tell me when you see it. Top.
Yes, yes. Yes, we can see. We can see the text.
Okay, very good. That's quick, so it's good. Okay.
So we shall wait till 2 p.m.
Merci beaucoup. Merci. Sorry?
So we shall wait till 2 p.m.
Yeah, 2 p.m. That means in
10:30 there.
10 minutes.
Yeah.
Okay.
Okay. Okay.
2 p.m. for you. It's 10:30
yeah,
in the
2
morning.
p.m. for us. Yes.
Okay. So in 10 minutes. No, you are in Kerala. Yes, I had a good friend in Kerala.
Uh-huh.
Professor Subramania Ranganathan.
Uh-huh.
He's a very famous chemist. Are you a chemist?
Yeah.
And you might know him. He died about 3-4 years ago. Ranganathan, he was then he was in Kanpur and he was in Hyderabad.
Okay, fine. Okay, fine.
Okay, so just wait.
We shall wait.
How will it start? Is there somebody making an introduction, or is it?
Yes. One of our faculty is comparing. First, first the principal of the college will be welcoming you. Then a faculty will be introducing you. Then we can start the lecture.
No, you tell me. I'm relaxed. I have done that many times, so it's the same thing all the time. Okay, very good.
Okay. So at the end of the session, we will take some questions from the audience also. Is it okay with you, sir?
To do what?
To answer some of the questions from the participants.
Get it.
Is it okay with you if some of the participants ask some questions or queries?
Yes, but it's better to have somebody filter the question. That's what I usually do. They should ask a question, and then somebody from the staff should filter the questions.
Okay, fine. Okay, fine.
Because it depends how they speak English, and so it's better that somebody from the staff does something like that.
Okay. We will read out the questions. Okay, fine. Admit them.
I think you don't need to talk so close to the microphone. One hears very well.
Okay, fine.
Sound is better when you're not so close. Okay.
Fine.
I have to take off my phones.
No.
Hello?
Hello?
Who are you? What is your name?
Manju.
Ajit Joy.
No, I'm actually Manju Kurian. I have to rename it actually.
I see. Yeah. Okay. How much chemistry do you have in your college?
8 of us.
How much chemistry? How many?
8 faculty. 8 faculty in the Department of Chemistry.
8 faculty. Okay.
Yeah.
How far is this Kottamangalam from Trivandrum?
Some 250 kilometers.
Huh?
250 kilometers. You have been to Trivandrum?
No, I was invited twice, but I couldn't go. I was too busy.
Oh,
And
okay.
maybe sometime later. Supposed to be pretty nice down there. Huh? Supposed to be quite nice in Kerala.
Yeah.
Okay. Another 5 minutes. Where are we here? You are in the chemistry department right now?
Yes. No, we are in the computer lab actually.
The computer room. Computer.
Yeah.
Okay.
Yeah.
Yeah.
So that we have uninterrupted supply, net connection. We are here.
One of my Indian collaborators just went back to India. He's now sitting in Delhi on quarantine. He's from Bhubaneswar. I see people have masks here, yeah?
Yeah.
I see
We are
somebody.
wearing masks.
Yeah. Now we have two, but not in the office. Because there's nobody coming into the office.
Here officers are working.
Sorry?
Here officers are working with 50% of the staff.
They are working, yeah? Working?
Yeah.
Yeah, yeah.
Yes. Yes.
Yeah, it's normally open, except that everybody has masks when they are not in their own office. It's quite a terrible, disruptive, very disruptive, this thing. You see the pointer also on the screen?
Yes, it's visible.
It works very well this morning, afternoon for you, because it's very quick now. I had some problems earlier in India and in China. There were too many people probably hanging on to it, and then it's slower. But this work today works very well. Now we have two other meetings in India.
Good morning, sir.
Hello, good morning.
I am Binu Varghese from the Department of Chemistry, Marathanesius College.
Okay, yeah. And what is your chemistry doing? What type of chemistry are you doing?
I am doing inorganic chemistry.
Inorganic?
Yeah.
Okay.
In complexation.
Complexation, yes. There will be a little bit of that. But it will be a very broad lecture because you have people from many departments and different origins, not only chemists. Yeah?
Okay, sir. Yes, sir.
Are there also people connected from other places?
Yes, sir.
Which places? Where from? You know?
In different parts of India in this webinar.
Yeah, this is now quite common. People
Yes.
have organized a seminar at one place and then many others connect. But where are they coming from, the ones connecting to you here? Mostly from Kerala or around Kerala, Tamil Nadu or something like that? Huh? Hello? Hello?
Okay, sir. We will start the session in 2 minutes.
Do you hear me well?
Yes, yes, sir.
Okay.
Actually, it is not very clear.
Who will be starting? Who is starting? Who?
Sir, shall we start?
Okay, go ahead.
Sir, shall we start?
Yes, sure.
Good morning, Professor Jean Mary LEHN, and good afternoon all. Hearty and jovial welcome to Nobel Laureate Lecture Series, Lecture 3, Steps Towards Complex Matter Chemistry. Organized by the Postgraduate and Research Department of Chemistry, Marathanesius College, Kodamangalam, Kerala. We are extremely enchanted and thrilled that our previous Nobel Laureate lectures was well appreciated and well acknowledged. Marathanesius College is a debut and premier institution in the field of higher education in India, accredited at A+ grade by National Assessment and Accreditation Council, sanctioned with UGC Paramarsh and DBT Star College scheme.
You should ask the people to turn the microphone off. There's a lot of noise. Turn the microphone
Yes, sir.
off. Other people should stop.
Participants, please mute your microphone.
That's better. Thank
Please
you.
mute your microphone.
That's good now. That's good.
Welcome back. Yeah. The Department of Chemistry regularly organizes conferences, seminars, and workshops in frontier areas of chemistry. Nobel Laureate Lecture Series is anticipated to stimulate the academic community towards research during this COVID-19 pandemic period. Keeping this in mind, we approach Nobel laureates in chemistry for a webinar. Today, we have an esteemed personality, Professor Dr. Jean-Marie LEHN, University of Strasbourg, France, to take us to the magnificent and astonishing world of supramolecular chemistry. Our admirable guest was fanatical from the very beginning and readily consented to our humble invitation. We are extremely grateful and privileged to have you, sir, to deliver a talk on steps towards complex matter chemistry. I know you all are impatiently and eagerly waiting for the much anticipated talk. Let us start the session by invoking God's blessings. First of all, I invite Dr. Shanti Avida, Principal, Marathanicius College, to welcome everyone. Over to you, ma'am.
Thank you, Dr. Binu Verghese. Am I audible? Am I audible?
Yeah, sure.
Yes, ma'am.
Very well.
Yes, ma'am.
Okay. There's most honorable Professor Jean-Marie LEHN, dear organizers and participants. Heartiest greetings from Marathanicius College, Kodamangalam. Marathanicius College, Kodamangalam is a nationally accredited A+ grade institution in Kerala, India, established in 1955 under Marathanicius College Association. The institution collectively nurtures the belief that education is a significant medium to effect a social transformation in young generations. Therefore, the focus is chiefly on academic competence combined with the social responsibility so that our students can contribute towards nation-building exercise. The college is now, in its way, scheming various strategies to be adopted to impart world-class education to students amid this COVID-19 pandemic. And this Nobel Laureate Lecture Series is thus a landmark achievement of the college in its pursuit towards excellence. I thank Professor Jean-Marie LEHN for accepting our invitation and spending his valuable time for the inspiring minds in the field of chemistry. On behalf of the students, staff and management of Marathanicius College, Kodamangalam, and everyone present here, with immense pleasure, I honor Professor Jean-Marie LEHN and offer him a warm welcome. Also, I congratulate and thank the organizing committee, the Faculty of Chemistry, especially Dr. Manju Kurian, Dr. Binu Verghese, and Dr. Gemma Francis for their efforts to make this event happen. I wholeheartedly thank Dr. Binu Verghese, our Honorable Secretary of Marathanicius College Association, for the encouragement and moral support for the lecture series. Dear participants, have a marvelous and engaging hour with the great scientist. Welcome and thank you once again.
Thank you, ma'am. Now I invite Dr. Densily Jose, Associate Professor in Chemistry, to introduce our guest of honor. Over to you, ma'am.
Am I audible?
Yes, ma'am.
Good morning, Professor Dr. Jean-Marie LEHN and dear participants. We are here today for the third lecture of the Nobel Laureate Lecture Series by the eminent scientist, Professor Jean-Marie LEHN. The nature of the chemical bond is the heart of chemistry. Supramolecular chemistry, the chemistry beyond the molecule, whose goal is to gain control over the intermolecular non-covalent bond, covers the structures and functions of the entities of higher complexity than molecules themselves. It is a highly interdisciplinary field of science and technology, bridging chemistry with biology and physics. Professor LEHN received Nobel Prize in Chemistry in 1987 for the development and use of molecules with structure-specific interactions of high selectivity. Professor Jean-Marie LEHN was born in Rochum, France in 1939. In 1970, he became professor of chemistry at the University Louis Pasteur in Strasbourg, and from 1979 to 2010, he was professor at the Collège de France in Paris. He is presently professor at the University of Strasbourg Institute of Advanced Study. He shared the Nobel Prize in Chemistry in 1987 for his studies on the chemical basis of molecular recognition, which plays a fundamental role in biological process. Over the years, his work led to the definition of a new field of chemistry, which he has proposed calling supramolecular chemistry, as it deals with the complex entities formed by the association of two or more chemical species held together by non-covalent intermolecular forces. Subsequently, the area developed into the chemistry of self-organization process and more recently into constitutional dynamic chemistry. Author of more than 950 scientific publications, Professor LEHN is a member of many academics and institutions. He was visiting professor at other institutions, the ETH in Zurich, the University of Cambridge, Barcelona, Frankfurt. He has also received numerous international honors and awards like Gold Medal of the Centre National de la Recherche Scientifique in 1981, Carl Ziegler Prize in 1989, Davy Medal of the Royal Society in 1997, Laureate Medal in 1997, Knight Commander's Cross of the Order of Merit of Federal Republic of Germany in 2009, and Grand Officer of the French Legion of Honor in 2014, to name a few. Being the president of the non-governmental organization called International Organization for Chemical Science in Development, he contributes immensely to help chemists in developing countries. Professor LEHN also studied music, and it became his major interest after science. He has continued to play the organ throughout his professional career as a scientist. So it's rare and great opportunity for all of us to meet and interact with such a distinguished personality. With great pleasure, on behalf of Martha-Ignatius College and all participants, I invite Professor Jean-Marie LEHN. Over to you, sir.
Okay, should I start? You hear me?
Yes, sir. I can hear you, sir.
You hear me?
Yes, sir.
Oh, very good. So first of all, thank you for the invitation to participate in this series of lectures. And it's always nice to have a new community and been talking to students. I've never been in Kerala. I was invited twice, but I could not go at that time. Hope maybe in the future sometime. I've been many times in India, in other places. So first of all, I have somewhat modified my title, but the content of the lecture is not very different. And I would like to tell you about a journey towards complex matter. And of course, life is complex matter. And this involves chemistry, molecular, supramolecular, and the field we now are mostly working on, which we term adaptive chemistry. You will see at the end what that is. But initially, I must start very early on in the life of our universe. In fact, right at the beginning. After the Big Bang, here, the universe was extremely hot and it cooled down very quickly. But there was no chemistry. That's the age of physics. No chemistry at that time. When the universe cooled down, progressively particles formed, and out of the particles, then atoms could form. And when atoms formed, they could be condensed together, they would react with one another and generate molecules. And that's where chemistry started, about 300,000 years after the Big Bang. That's the age of chemistry. Of course, physics and chemistry are continuing all the time, but something new happened, at least on our planet, Earth. A new property appeared in these complex chemical entities. It is life. Life started on Earth about here, and this is the age of biology. But it did not stop there. It continued to evolve, and the organisms on our planet developed another property, which is called thought, thinking, represented here by a very famous sculpture by a French sculptor, Auguste Rodin. And this is steps towards complex matter or steps towards life, if you want. So, first question is, of course, that the universe doesn't stop at present times. There are many people around the world who would like to stop things. They are totally unable. The universe does not worry about human beings. The universe will continue to evolve on the basis of its laws, the general laws which rule our universe. And this will continue, and nobody will be able to stop that. So what we can do is to try to live as well as possible, but stopping the evolution of the universe is, of course, totally impossible. Furthermore, I would also like to insist right now that we are very, very probably not alone in the universe. Human beings are only a point in space, that means on a small planet called Earth, very small thing, and time, the times we are now on. This will continue, and there are other planets, exoplanets, where life can exist. Very probably there is life. So then we have to ask a question, what is in this universe? This is not a slide, a wrong slide. It's a dark slide. Why is it a dark slide? Because cosmologists tell us that there is in our universe 68% dark energy, 27% of dark matter, which means altogether 95% darkness. So that is not so interesting, but there are 5% left. And this 5% is what we call visible matter, what cosmologists call visible matter. This is the matter that matters. Why? Because we are part of 5% visible matter in our universe. The rest is 95% dark energy and dark matter. So we are very little in our universe. Now, matter evolved as a function of time by becoming more and more complex, higher and higher complexity, as you see here. So at the beginning, it was divided, particles. Then it became condensed, particles uniting to make atoms, molecules. It became organized, then living. Matter became thinking. And beyond that, there was maybe something even more complex, but we don't know what it is, but we cannot exclude it. And this is the evolution towards complex matter as an evolution towards more and more information in some places. I insist on that, just in some places in the universe. Now, in order to understand what is going on, we have to ask a big question. What I consider as probably the biggest question asked for us, for our activities, our scientific activities. The big question is, how does matter become complex? What are the pathway maybe, by which matter can become more and more complex? And of course, this is a very important question, and mankind invented something which we call science to give an answer to this question. how did we go in our universe from an elementary particle to a thinking organism, and maybe to higher states of complex matter than the ones we know about. So, as I said, humanity invented, created science. I will consider only three main domains. First of all, physics. Physics deals with the basic laws of the universe, those on which everything else depends. Please switch off your microphones. Switch off your microphones. Switch off your microphones. Biology studies the rules of life, and chemistry is building the bridge between the two. Chemistry tries to discover the ways in which one can go from general laws, the general laws of the universe, to living organisms, to thinking organisms. So chemistry has an extremely important role, not to discover the basic laws, but to see, to check, to understand how basic laws can lead to specific expressions of our universe, which are, for instance, human beings on the planet Earth, and very probably other beings on other planets around the universe. So the answer to that question, we can give a name to it. Matter becomes complex, becomes complex by self-organization. That simply means that it is a result of the basic laws of our universe. That is what it is about. Self-organization is the driving force of our universe. It happens that our basic laws are such that our universe will become self-organized. We can even claim that it is a cosmic imperative, and that self-organization, organization in our universe will occur somewhere. It is not an accident. So for me, life is not an accident, not something impossible elsewhere. There is probably life in many places in our universe. This self-organization can be on the big scale, the cosmic scale, where gravitational forces operating on initial inhomogeneities in density and rates of expansion of matter at very early times, of the universe at very early times. This led then to cosmic structures, planets and galaxies and so on. But this is not what we are interested in. We are interested in self-organization of complex matter, which happens through the action of physical forces, electromagnetic forces, on the atomic and molecular matter and combinations, which result from the operation of electromagnetic forces on atoms, molecules, and by random structural combinations. So the chemical evolution of matter started with prebiotic chemical evolution before life existed on our planet. This is the self-organization of non-living matter. It is preceding the biological Darwinian evolution, which is in fact also a chemical evolution. So chemical evolution is much broader than Darwinian evolution. Darwinian evolution is an evolution of living organisms on our planet. On the other hand, Darwinian evolution is also a chemical evolution. Darwinian evolution is also a chemical evolution because it is due to the way in which the genome, the information of living organisms, works, and this we will see again later on. So chemistry can be defined as the science of the structure and transformation of non-living and living matter. But we will see later there's another property of chemistry which we have to take into account, which is very important. So structure, that means how matter is built, And transformation, how can you transform matter between different entities? So chemists over the years, then for many, many years, analyzed matter around us and came to the conclusion that there were many things, many sort of units, entities, which they called elements. And then this was sort of understood. People discovered many of those elements, and they tried to organize, to understand. Initially, this was quite chaotic. There was no organization until the middle of the 19th century, when a number of people, the most famous of them, Dmitri Mendeleev, proposed that matter is formed of particles which can be organized, and these particles are the atoms. So in 1869, Dmitri Mendeleev published a paper which I consider as one of the most important papers in science. From physics to biology, one of the most important papers in science, I insist. And here is the paper published in 1869 in a German journal called Zeitschrift für Chemie. In English, the title of the paper is On the Relationships Between the Properties and the Atomic Weight of the Elements. Mendeleev had noted that using the properties of the elements and their atomic weights, one could organize them in a table. The notes that 2019 was the 150th anniversary of this very, very, very important publication. And this is here what you see, the initial organization of the elements proposed by Dmitri Mendeleev in this paper. They were nicely organized, as you see here, beryllium, boron, carbon, nitrogen, oxygen, fluorine, and so on. You see here sodium, potassium, rubidium, cesium, and so. Mendeleev went even a step further. There were some places where he put question marks here and here and here. Why did he put question marks? Because this was, so to say, an element which was lacking in the regularity of his table, and he predicted that they would be discovered. And of course, they were discovered. Nowadays, this is called the periodic table of the elements, and this is the present way it is represented. Now, what you have before your eyes is one of the most important of all progresses of everything done in science. Remember that, what I said, I insist. This is one of the most important discoveries of science and of the science called chemistry. So chemistry has made a most important contribution to the knowledge of matter. Why? Because what you see here are the bricks of matter. The bricks of matter which make up all visible matter everywhere in the universe, not just on the planet Earth, everywhere, because these elements are forming a table which is complete. And why is it complete? Because it's just a question of looking at the structure of the corresponding atoms and counting. Hydrogen is 1 proton in the nucleus, 1 electron around it. Helium, 2 protons, 2 electrons. Lithium, 3 protons, 3 electrons. You just count 1, 2, 3, 4, 5, 6, 7, and so on, until uranium, 92, which is a natural element. The other ones, these are synthetic ones. So, what does it mean? It means that this table, I insist again, is a table which represents the bricks of visible matter everywhere in the universe, on the planet Earth, or millions of light years away on other galaxies, other planets, and so on. And I think it's quite incredible that this could be discovered, could be found, and that we now know what we will find everywhere else of visible matter. These are the elements of the universe, not just of our planet. And this is a chemical feat. That is what chemistry brought to science, the most important thing. Now, this is the playground of chemistry. Chemists are like children. They use their pieces, their elements, and connect them together, making complex entities, modifying them, transforming them. And this is basically, put simply, the work of chemists, making new forms, or understanding the forms of matter and making new forms of visible matter. Now, what it is made of is fine. We need to know that, and we just saw it. But as chemists, we also want to understand how we can transform, how chemistry can transform compounds into one another. And that is based on the chemical reaction, which was the equation of the chemical reaction was formulated by Lavoisier in the 18th century, when he said, nothing, rien ne se perd, nothing is lost, rien ne se crée, nothing is created, everything is transformed. And this is exactly what chemists is. What chemists doing in the laboratories is to transform compounds into one another. And this is a very powerful activity which enables chemists to transform matter, objects of matter, into one another. Now, chemistry, therefore, has the basic building blocks in the periodic table and the transformations. But is that all? Are chemical objects just made of elements, connecting elements? And is this just a graph, a sort of a graphical representation? That's not the case. In fact, in 1874, two chemists realized and proposed and published independently that chemistry deals with architectural objects. That means objects which have a shape. It's not just a set of elements connected in a given way, but these elements, these connections, these molecules have a shape. And this was the realization that chemistry was an object in space, three-dimensional space. It was the birth of stereochemistry in 1874. The two chemists were Jacobus Henricus van 't Hoff in the Netherlands and Joseph Achille Nobel in France, in fact, very close to Strasbourg. So this was a major step, of course, to understand how chemical objects are built. So let me now, from there, go on and consider that, of course, I could talk a lot more about what happened in chemistry, how it is transformed, how it has been building up, but let's not summarize in some ways and say from the atom to the molecule, chemists have been building molecular chemistry, the way in which one can connect atoms to form complex molecules and transform these molecules into one another. This chemistry is mastering the organization of visible matter, visible molecular matter, by using strong binding interactions between the atoms which form these molecules. So this molecular chemistry developed enormously, and it's a very strong field. And let's look at two milestones. The first milestone deals with molecular chemistry in the 19th century. In 1828, a German chemist, Friedrich Wöhler, made urea from ammonium cyanate. Now, what does that mean? This means that in the laboratory, Wöhler has been able to transform what is called ammonium cyanate into a compound called urea. This urea, what is going on here? Who's putting these dots on the screen? Could you remove that? What are these things here? So Wöhler showed that he could, in the laboratory, make urea. Urea is a compound contained in living organisms, and that means that he was able to generate in the laboratory a compound which is part of living organisms. This was a way to destroy the idea that for making anything contained in living organism, you needed a magic force called vital force. There is no magic force. There is no kind of effect like that. There is no entity in the universe using magic forces. There is science. And Wöhler showed that you can make urea from a non-living, from an entity not present in living organisms. Now, 150 years later, a major breakthrough was the production, the synthesis, this building up in the laboratory of these very complex molecules called vitamin B12. This vitamin B12 molecule is, of course, much, much more complicated than urea. So it was a major enterprise, and two groups collaborated in the building up of vitamin B12 from its elements. Robert Burns Woodward at Harvard and Albert Eschenmoser and his group in Zurich. So both groups collaborated, and in the beginning of the 1970s, this synthesis, this build-up of this molecule was completed. It is very complex, and it shows, however, how strong the knowledge of chemistry has become, how strongly people were able to build up complex structures of matter. Since the 1970s of last century, of course, the 70s of last century were not the end of molecular chemistry. It continued to evolve. And today, of course, many new reactions, many new have been discovered, many new transformations, many new compounds have been made, drugs, materials, and so on. And this will continue. However, we can ask a question. Besides molecular chemistry, which is a very adult, strong science, continuously developing, what else should we think about? Let me illustrate that. Here is a cancer cell in blue and two killer cells in violet, purple. These killer cells must find out if a given cell has become cancerous, has been a cancer cell. And if they find a cancer cell, they have to destroy it. But how do the killer cells here know that the other partner, the other unit, entity here, is a cancer cell? What tells them that it is a cancer cell? If they make a mistake, there is a big problem. Then, of course, maybe they don't destroy a cancer cell, or maybe they destroy a healthy cell. No good for you. So something must tell the killer cells that the other one is a cancer cell. Similarly, if we look at a white blood cell, artificially colored in rose, these blue dots are the HIV virus. When this HIV virus encounters a white blood cell, it can infect it. How does the virus get into the cell? How does, so to say, the virus know that it can now enter the cell, infect it? Something must happen between the cancer cell and the killer cells, between the white blood cell and the virus particles, which has the information about each object. Now you well recognize that this object, this cancer cell, is made of a membrane which contains molecules. These killer cells are also made of membrane which contains molecules. And obviously you realize that it's by touching that something tells the killer cells that the other one is a cancer cell. It's by encountering, by hitting, by attaching to the white blood cell that the HIV virus sort of knows it. The virus doesn't think, of course, but the virus gets the information for infecting the cell. This leads to the idea that something must happen between the molecules which make up these objects, these large cells. And indeed, that means that beyond molecular chemistry, there must be a chemistry which may be called supramolecular chemistry, lying beyond the molecule, and which relies on the other type of interactions, others than the atom-atom bonding in the molecules and the non-covalent bonds. The basic processes studied over the years were how do molecules recognize one another. This is very basic, very important, for instance, for killer cells to recognize cancer cells. How do chemical reactions occur? How does transport occur between a membrane where then some entity helps another one to go through a membrane? I will come back to that. So molecular recognition is the basis process, which is, so to say, the most important one at the basis. How can we define molecular recognition? Of course, it's a complicated, a complex process, but we can simplify it at least at a given level. First of all, recognition requires interactions for binding. The molecules have to bind to one another to be able to perform some recognition. If there is no interaction, of course, they ignore each other. But very most importantly is another property, which is these processes involve information. You cannot have information. You cannot do a recognition process without information. So this is a very basic property of chemical objects, and I come back to it. So how can we sort of very simply present molecular recognition? We may call it as a sort of a complementarity between the geometry of the objects in interaction and interactional features. And in the 19th century, there was already a very good idea, a very geometric one, but quite a good idea about how molecular recognition may function. And that was due to a chemist, a German chemist, Emil Fischer, who in 1894 published a very important paper where he said for an enzyme to act on its substrate, they have to fit together like Schloss und Schlüssel, like a lock and a key. This is a very mechanical representation of molecular recognition, but basically it fits. It can be considered as a very important step to understand molecular recognition. Emil Fischer got his doctorate at our university in 1874, and of course we are very proud of that. Now, you also noted that the 19th century was extremely fruitful for chemistry, and chemistry was the science which sort of let people understand matter, how it behaves, how is it composed. So now, what is the next step? Understanding molecular recognition. And what is the most important process in molecular recognition? It is arguably the genetic program which is written, the genetic information which is relying on molecular storage of information in an entity which is called the double helix of DNA. Let's analyze that. Of course, most of you must know about this, but it's worth looking at it. The double helix of DNA and the storage of this molecular information, of this genetic information, is by attaching chemical groups here on a long chain of rather simple composition. So the order, the sequence of these letters represents the genetic information for a living organism, and what changes is just the order of these 4 letters. We note as chemists that these 4 letters here are chemical groups which are rather simple, in fact. So what makes the difference between a tomato and an elephant is simply the way in which these 4 letters are organized along this long strand. And then these strands, 2 such strands wrap around each other and form the famous double helix, which you see on the right-hand side here. The question is then also, if that is the storage of molecular information, how can you read this information? It can be read out by simply pairing up the letters. One letter, adenine, can pair up with thymine or uracil derivative, and by 2 points of interaction represented by these dots. Guanine, another, the third letter, and the fourth letter, cytosine, they interact by 3 points of touching, of pairing up, represented here. So it is a binary system. Molecular recognitions occur through complementary interaction patterns, 2 supramolecular bondings, and 3 here. It is a binary system, 2, 3, 2, 3, 2, 3. And this is now by interaction between the molecules, these molecular groups. Therefore, it's a supramolecular process. We may therefore say that chemistry is an information science. It's a science, of course, of the structure of matter, of the transformation of matter, but also of information in matter. The storage of this information is in the molecule at the molecular level, and the processing of the information is by contact between the objects at the supramolecular level. So chemistry is the science of informed matter. Now, let me just tell you about my initial motivations, how this field started. I was initially interested in neurochemistry, the chemistry of the nerve system. And I was looking for a process which would be rather simple. This was in 1960s of the 60s of last century, a long time ago. And the question was, what are the processes which one might start with to try to understand? I considered that in fact the propagation of the nerve influx is a process which we might try to understand. This propagation of nerve influx rests on the formation of an action potential, which comes from exchange of sodium-potassium against the nerve membrane, and which leads the signal along the axon, along the nerve, the axon of the nerve. So sodium-potassium, these look like rather simple objects. And the conclusion was, for me, that there must be molecules in these nerve membranes which can make the difference between sodium and potassium. The result was that the question asked is that there must be selective binding and transport of sodium and potassium ions through the membrane of the axon. Question then, how is it possible to selectively bind sodium or potassium ions? These 2 ions are part of a sequence, one of Mendeleev's columns. These Mendeleev columns are the alkali metal cations. They are spherical chemical entities which bear 1 positive charge and increase in size from lithium to sodium to potassium to rhodium to cesium. And 2 of them are very important, especially important. They are the sodium and potassium ions, which make up the action potential. So the question I asked myself was rather simple. How can we make in the laboratory objects which will selectively recognize sodium and potassium, and of course then also extend it to the general binding and transport of spherical ions, which means understanding spherical molecular recognition. We then, in 1968, prepared in the laboratory a selection of molecules, which we called cryptans, because there were molecules with a cavity, a crypt inside. By chemical synthesis, one can modify the size of this crypt and adapt it so that they can form inclusion complexes, cryptates, with spherical ions. The smallest one corresponds to lithium, the second one to sodium, the third one to potassium. So this was a change, an adjustment of cavity size to sodium, sodium, potassium, and so on. And therefore, it was a way, it was discovering and studying spherical recognition, how we could get very strong spherical recognition. At that time also, some natural antibiotics were shown to bind potassium, for instance, and of course the ground ethers of Peterson were also developed at that time for binding lithium, sodium, potassium, and other ions. So that was the start. I don't want to insist more on it, but it was the start of molecular recognition studies, and they were sort of used the idea of Emil Fischer. At least you can interpret it in that way, that you make locks for the keys, lithium, sodium, potassium. And the locks here have a shape which corresponds, which is complementary to that of the key, which goes into it. So molecular recognition is the process by which you try to build up a synthetic receptor molecule, which then would bind one key among a set of keys, and here the red one, which means this is a sort of a Emil Fischer type molecular recognition process where a given lock recognizes a given key and binds it. So very numerous studies have been performed on molecular recognition process everywhere around the world, and of course, very many in Germany. So I have many colleagues and friends there who work in this area. So let me just summarize at that stage. Chemistry has been defined as molecular chemistry in 1828 by the work of, we have just seen it, Friedrich Wöhler. And this led to chemical synthesis, which is able to build up synthetic receptors, so that in 1978, I proposed the name of supramolecular chemistry for the field, which deals with the way in which a receptor can bind a substrate through non-covalent interactions and make a supramolecular object, which can have properties such as recognition, transformation, transport, or translocation, and therefore build up functional supramolecular systems. This type of activities have been going on in many laboratories, not just recognition, but also transformation of chemical objects and transport through membranes, recognition processes, reactivity processes, and transport processes. Let me just also mention a few of the applications in life sciences. First of all, you realize very well that, of course, basic research is acquiring knowledge. Applied research is applying knowledge, so applications are important consequences of new knowledge. First of all, molecular recognition is basically important for drug discovery. Why? Because a drug, a biologically active substance, is a chemical key for a biological lock. So the processes of lock and key, complementarity and binding are basic to drug discovery. Secondly, we also made a number of other derivatives of these cryptates, and here there is one which has an interesting property. It binds the europium ion in its cavity, and light can be absorbed from the organic skin, building up this cryptand, and then the energy transferred to the europium, which then shows a strong red luminescence. This type of object, this type of nanobulb, so to say, has been used to develop an immunoanalysis system, which is used in many hospitals for various medical diagnostics. The transport processes, translocation, transport processes have, of course, play a very, very important role in biology. They also play a role in biotechnology and in gene therapy. In other words, the transfer of a gene into a cell is a supramolecular process whereby a synthetic vector binds to a piece of DNA, a gene, and helps it to go through the cell membrane to go to the nucleus, make its RNA, and then being transformed into this protein. So this is a very important process, and it allows to, first of all, act to develop, hopefully, therapies for genes, for diseases which are caused by a gene which does not function properly. It is also very important for biotechnology to make genetically modified organisms. Now, here I would like to insist on one thing. People are nowadays, not all, but a number of people are afraid of GMOs. I guess that is totally absurd. This is totally stupid. GMOs, we need them, and we will use them. The fact that some people protest against GMOs is totally wrong. GMOs are necessary. They will enable us to control living systems and to make new types, for instance, of plants, which are, just to give an example, very important for food. Then there are also supramolecular materials which have been developed. They can be biomaterials, and I would like to show you two cases of supramolecular materials, which are of quite general interest. First of all, because supramolecular polymers, they are polymers of supramolecular bases, can be transformed into films. These films can be made, you see here one, a very nice film of a supramolecular polymer where there is hydrogen bonding between polymeric chains. So this was made, you see two fingers here, these are the hands of one of my Indian co-workers, Dr. Nabarwan Roy, when he was in my group, and he made this film, which you can cut with scissors. And then you can superimpose the two ends. You press then with your finger in the middle here, where they are superimposed, and they stick. You can pull and they stick. So the material has undergone self-healing. The cutting has been healed, and that is one property of supramolecular polymers, which is a very interesting one in the future, and many people are working on it. Another interesting property was the following one. Supramolecular materials can also be transformed, made as biomaterials, biocompatible supramolecular materials. And these supramolecular polymers are biocompatible. And in 1990, we had proposed in a paper at that time, 1990, the concept of supramolecular polymers. And then it developed quite well, thanks to the work of many groups around the world, especially, for instance, Bert Meyer in Eindhoven in the Netherlands. And we worked quite a bit on it too, until in 2013, I was informed that a small company, Xeltis, had developed biocompatible supramolecular polymers for cardiovascular implants. Supramolecular chemistry, which could be used for the reconstruction of the heart of children who had a severe cardiac malformation. This was quite a breakthrough, and it was in 2013 used as such for the surgical treatment of children. Note that it's 23 years from the initial proposal of supramolecular polymers to the activity, the implementation leading to medical use. And here is the first little girl, Dominica, who on October 23rd, 2013, was implanted with this cardiovascular, in her heart, the introduction of a supramolecular polymer to correct for this cardiac malformation. This was done in an institute, in a scientific center in Moscow for cardiovascular surgery by Professor Leo Bokiria, you see. This was, of course, factory to see that these compounds could also help children and human beings in general, of course, and therefore contribute to health. More than many children now have been operated on by introducing either patches or conduits in their heart. And the same company has now developed also pulmonary heart valves, which have been implanted in children in Budapest, in Krakow, and Kuala Lumpur. So a lot of progress has been made, and it continues. And the fact that we have also been able to contribute to the health of human beings, and especially in this case to children. So let's go back to the main theme I have been, which is the question of, can we, on the basis of what we know about supramolecular chemistry here, can we, on this basis, also develop more systems which can self-organize? As I said earlier, self-organization is the driving force in the development or in the organization of visible matter in the universe towards living systems and towards thinking systems. So can we use molecular recognition to set up in the laboratory systems which can self-organize? That means spontaneous but information-controlled generation of complex supramolecular architectures via molecular recognition patterns. Let me just show a chemical process which does that, but which is present in nature. This is the way a virus builds up. The tobacco mosaic virus has a coat which is made of 2,130 pieces, bricks, protein subunits, which I show you here. This is an extension. This is the more condensed picture of it. And this is even more simplified, where this piece here, which is like a piece of pie, can assemble and form this column. And inside the column, the RNA, the genome of the virus, will be present, will bind. So this is a chemical process. It has nothing to do with life. It's just a bunch of molecules which go together in organized fashion, in fashion which is organized by the way in which they interact surface to surface. It's a sort of a self-recognition because one side of the molecule recognizes the other side of the next molecule next to it. And that is a spontaneous process. It is not magic. We understand all the basics of it. So it looks like a programmed chemical system, yes. And of course, the infamous present virus, this SARS-19, COVID-19, this one is just the same thing. It's built up by interactions between the molecules, between the proteins which form the virus. And we have to find a way to stop that by chemical or maybe biological means like vaccination. So these programmed chemical systems rely on a molecular program where information is stored in the components and on the supramolecular processing, supramolecular operation, where the interaction patterns lead to the processing of this recognition information. Let's have a look at how this can be translated into processes which you can implement in the laboratory. So we shall look at metallo-supramolecular architectures. That means architectures resulting from the binding together of molecular bricks, ligand molecules as bricks, bound together by a cement connections, which are metal ions. Let's see what can be done with this. Of course, we know very well that inorganic chemistry is a combination of metal ions. And this, of course, leads to the design of ligand molecules capable of leading to metal cations, of using metal cations to build up. there's somebody playing with the screen. Could that person stop, please? Could you ask the people who are playing with the screen and making drawings on the screen to stop doing that? So many self-organized metallo-supramolecular architectures have been produced. For instance, double helices and artificial double helices and triple helices, grids or grid-like objects where organic molecules are crossing, 3 vertical, 3 horizontal, and at each crossing they are held together by a metal ion, in this case silver. One can make nanocylinders like these components which can assemble 3 linear molecules, 4 flat molecules, and here 12 metal ions which hold them together. Altogether, 19 components which spontaneously assemble and generate this, which you may call nanocylinder. Many others have been made. Here are just 3 other examples of metallo-supramolecular architectures of various complexities which have been produced in the laboratory. So this type of approach can also be interesting for nanoscience and nanotechnology, because one can think of trying to make the components of devices and give them the possibility to spontaneously assemble so as to generate the final device. That would mean to, rather than to have to fabricate or to manipulate the components to build up the device, we would have a self-fabrication where rather than fabricating, we could then go to self-fabrication, which of course would be the ultimate fabrication. And I think in the future, this powerful means of supramolecular self-organization can be implemented to realize such self-fabrication of complex objects. For instance, what you might call a molecular computer. So I would like just to indicate now to you what are the present directions of what we are doing. I will simplify it and then go to some general conclusions. I have tried to show you up to now that chemistry can be done by designing its objects, introducing information and programmation so as to be able to have to generate a controlled formation of functional molecular and supramolecular entities. This has been done and is very much many activities. It's very much pursued now in many laboratories. There's another step. The next step is, why not let the system select what it needs to build itself up? This selection requires that you have different type of objects. That means a diversity of objects, of bricks, and dynamics. That means that the construction will be able to dissociate and reassociate and change itself so as to be able to change its constitution, to make the dynamic, to give dynamicity, to make the chemical object dynamic, meaning that it can dissociate and reassociate and therefore adapt to different agents from the medium. And that leads to adaptive chemistry, which is now our main field of activity, adaptive chemistry. This adaptive chemistry, which relies on the constitutional dynamics, that means on the possibility that the constitution of the objects can modify itself through exchange of components and through expelling some or taking in others. And this would then lead to this property of having systems which can adapt by variation of their constitution on both the molecular and supramolecular levels. This has introduced and made possible in the last 15 years or so, maybe now about 20 years, let's say 15 years, new methods for searching for biologically active substances, trying to set up self-organization processes of dynamic nanostructures, and also developing dynamic materials, which would be responsive to various agents. And that leads to adaptive chemistry. Now, at this stage, in view of the time, I will simplify a little. I will go through some, but I would nevertheless just like to show you what can be done, even without explaining exactly to you how it is doing, how it's operating. First of all, for looking for biologically active compounds. Suppose you have components, shown here on the left, which can interact by this black dot and build together. If you mix them, they will generate these constituents, which are all the possible combinations of the components. So what can you do with this? You can use the principle of mass action, the Chatelier rule. And this means that if you introduce a receptor to this mixture, which can always reorganize itself, it will, out of these possible here units, these are only some of them here, many others may be formed, they can select the one which fits best, the one which has the red piece, the green piece, and the yellow piece. These three pieces are the ones which fit best, so they will interact, and they will therefore represent, so to say, a lock which is assembled by its key. Selection and amplification of the best complementary entity, the lock assembles its keys, and this can be used for the selection of the best binders and for various biological type of processes, and including drug discovery. Interesting also is the fact that you can make entities which are films like this one, a nice, very nice film, which contains imine bonds, which are reversible bonds, and also pieces which can be biodegraded, so that this is a double disintegration dynamic polymer, and this kind of film is of interest for making degradable plastics, degradable here for two ways: cleavage of imine bonds by water, that's chemical degradation, and biodegradation of the units, which are here in the middle. I go quickly here, as I said. These are, so to say, green plastics. Then one can also try to use different films and superimpose them, so that if this connection here is dynamic and can break, it can then reconnect differently. Here you see A is connected to B, and A prime is connected to B prime. But if these things can break and reform these bonding units, then A can connect with B prime and A prime with B. Now, I am going fast, I know, maybe not all clear, but I want just to show you what can be done in this case. Let's look at two films which are connected, which are superimposed just here. And here, in these places, we can generate the new combinations which were not present at the beginning. A with B prime, A prime with B. And maybe one of those has a color or a fluorescence. If that's the case, what can we do? Let's see here. This was my Japanese co-workers, postdoctoral and doctoral co-workers, liked this nice picture, this small picture. That's a cat with the head and the ears being polymer AB, the film AB. Inside of the ears, eyes and mustache being the polymer A prime, B prime. And so they superimpose that the way it's done here, and then they heat it to see what happens. And you see color and fluorescence appears where it was superimposed. In other words, this demonstrates that at these places, one has a recombination of the components so as to generate the two new combinations which were not present at the beginning. And it happens that one of these two has this property of having a color and having a fluorescence. So this just demonstrates that these dynamic features indeed work. So at this stage, let me summarize the question of the development of chemistry in a very, very simple way, but I think very relevant. We start with molecular chemistry. We have to know how to make molecules, then we look at the beyond molecules at supramolecular chemistry, organized, and then dynamic objects, which can change themselves in a dynamic way. and become then adaptive. That's towards more and more complex forms of matter. And of course, living systems and thinking systems are even beyond that, going up here, going to living and thinking systems. Will happen in the future. For the moment, it's not yet there. Let me just make some general comments at this stage. So the essence of chemistry, as you may have understood, is not just to study what already exists, what exists around us, what exists in visible matter on our planet. That is, of course, very important. But it is not only to discover and understand this, it's also to create novel expression of complex matter. That means the book of chemistry must be written, not only to be read. Not just to be read, it must be written. And the score of chemistry must be composed, not only played. Of course, you have to play it. Like when you play music, you have to have instruments and can play the music, but you have to compose the score. You have to know what you want to play. So chemistry has creative power because it can do that. And again, this can be represented by a sculpture of this same French artist, Auguste Rodin, who shows here the hand of the artist expressing a sculpture out of a stone. And of course, the stone does not contain this. The stone does not contain this sculpture. So it's the hand of the artist which expresses that. And in the same way, chemistry can express out of the stones, the elements which are given to it, the possibility to create new objects which did not exist before. So chemistry may be termed the art of matter. This was something which had been, so to say, expressed, of course. At that time, this type of knowledge was not known, was not something, science hadn't progressed enough. But there was a famous person, Leonardo da Vinci, who has, in the 15th century, already expressed that very well. Note that it's a 500th anniversary of its death last year. He wrote a very important sentence. He was a scientist, he was an engineer, he was an artist. And of course, all of you know Mona Lisa, I suppose, the very, very famous, probably the most famous painting in the world. He wrote the sentence, "While nature finishes to produce its own species." This is bioevolution, and we are part of it. Man begins, that's very strong, using natural things. We now know what that is. These are elements of the periodic table, in harmony with this very nature. That means in harmony, in agreement, following the laws of physics. And then the ending is very, very strong, to create an infinity of species. So Leonardo da Vinci was courageous enough to write that mankind can create, in fact, by science, an infinity of entities which do not exist, which did not exist just in nature. Let me just bring a piece of mythology in who is playing around. Prometheus was one of the gods in Greece mythology, like you have your mythology in India. Prometheus stole the fire of knowledge from the gods and gave it to mankind. And he ran away, as you see here, and gave this power, this fire of knowledge to mankind. We must realize that we cannot give it back. That what we know, knowledge, we cannot erase. We cannot give it back. What we know, and we have to live with it. That means our path leads us from the quest of knowledge to the control of our destiny. We acquire knowledge, we will not be able to give it back. All these people who want to erase knowledge, This is totally impossible. They are foolish. So our path leads us from the quest of knowledge to the control of our destiny. And we will control our destiny in the future more and more. In the past, this transfer of information, of knowledge happened, let's say, in this way, for instance. Then in the present, it can happen through communication, through human beings. In the future, it may possibly happen in this way. I mean by that, not that we will only be shaking hands with robots, but it means that we will modify ourselves and have relationships with different individuals and maybe even robots at some stage. And we already have modified ourselves. Many people around the world have a new lens in their eyes made of polymers. That is not a natural lens. People may have in their legs some kind of a metal to fortify when the leg was broken. That is not natural, so to say. And even if you get a heart transplantation, that heart transplantation is not your heart anymore, and it may be an artificial heart. So we will modify ourselves and will continue to do so, and we will take power over what we are and not let it evolve by natural means, but we will do that ourselves as human beings. Let me cite a mathematician to finish. David Hilbert is a very famous German mathematician, which is buried here in the cemetery in Göttingen, a small university city in northern Germany. And there are two sentences written here. You can see what are these sentences. They are very strong sentences. The top and the bottom one, we'll have a look at them. The top one is, wir müssen wissen. We must know. This is already very strong. This is what science, what drives us scientists. We must know. We want to know and we must know. But the second part is even stronger. The second part, wir werden wissen. We will know. This is a vote of confidence. That is what let's say the driving force which pushes scientists to build up science. We must know, sure. And we are convinced that we will know. Through science, we will know. And since I expect that many young students are listening to my talk, I tell them science shapes the future of humanity. Participate. And in addition, for all these young people, I would like to say that science has another property. Science has no borders. For science, there's no dogmatism. There is no religion. There is no limit. Science breaches everything. That means around the world, there is science without borders. When you are a scientist, you have no borders in the world. And I would even say that human beings that other living organisms on on other planets will also be able to talk with us through science. Thank you very much for your attention. Hello?
Thank you very much, Professor, for carrying us to the exciting world of chemistry, and you have taken us through the milestones of molecular chemistry, especially in supramolecular chemistry, in a lucid manner. Now, it's the time for the interactive session. Professor LEHN will address some of the queries of the participants. The participant can post their questions through the chat box of the Zoom and YouTube live streaming. Sir, can I ask? Sir, Dr. Srinivasan KB has asked, is it necessary to have solvent atmosphere for supramolecule interaction to take place?
To have what? To have?
Is it necessary to have solvent atmosphere for supramolecule interaction to take place?
No, supramolecular interactions are basic physical interactions. They are present. It's like there are links between atoms. There are links between molecules, and supramolecular objects are just the result of interactions between molecular entities.
Thank you, sir. Can the same molecule act as host and guest system through terminal interaction?
What interaction?
Terminal interaction.
I don't
Can
understand.
same molecule act as host and guest system through terminal interaction?
Terminal interactions?
Yes, sir.
Yeah, sure. Supramolecular interactions are based on non-covalent bonding, and these physical forces are the ones which lead to the supramolecular object.
Okay, thank you, sir. Another question from Darshana Baburaj. Is there any harmful effects related to supramolecules in drug discovery?
You know, this is a question, of course, people getting more and more sort of afraid now with some dangerous things and so on. No, look, I mean, it all depends what we do with it. Supramolecular chemistry is a science, and what mankind, what human beings do with it, that's their problem. If they use it for bad reasons, then this is their responsibility. Drug discovery is what they're not. In fact, supramolecular chemistry should make and is making drug discovery easier, understandable, because, as I indicated, a drug is a molecular key for a biological lock. So the better you, the better you know how to build the key, the more you can make it selective for exactly that lock. So avoiding side effects. In other words, an understanding of molecular recognition makes drugs more safe.
Thank you, sir. Dr. Manju Kurian has asked, what is the relevance of supramolecular interactions in inorganic chemistry?
Of course, organic chemistry, supramolecular chemistry is part of is an organic chemistry one, but it also uses inorganic chemistry. So supramolecular chemistry brings together some areas and major areas of organic chemistry, how to build molecules, how to make them selective for a given purpose, and inorganic chemistry, which can contribute units which can link the organic molecules together. So I think supramolecular chemistry has this interdisciplinary aspect of organic, inorganic nature.
Thank you, sir. How are the latest trends in complex matter chemistry applied in creating cyborgs and humanoids?
Creating what?
Cyborgs and humanoids. Humanoids.
Well, sorry, what?
Humanoids.
Humanoid?
Yeah, yes, sir.
That means humans, yeah?
Yeah, humans.
Yeah, we are not there yet. I mean, we just modify ourselves. And as I said, we may already have lenses in our eyes, new teeth. We may have new sort of bones or replacement of bones. So all this will be the result of science. It is, of course, much of chemistry because it deals with materials, material science. Material science is also chemistry, but of course it's also physics. It has also a physical part, that's quite clear. So we will be able to do that, and We have just to learn how to do it and to be careful when we do it.
Thank you, sir. Another question from C. Srilada. Whether supramolecules has made their presence in petroleum-like molecules, which can be used as a fuel replacement.
It can be used as what?
Fuel replacement. Fuel.
Fuel?
Yes, sir. Fuel replacement.
Means fuel.
Instead of petroleum.
Yeah, sure, yeah. Yeah, but petroleum is a chemical also. Huh? So I think let's not dream. I mean, of course, nowadays we want to get away with petroleum, but we will use petroleum for quite some time. It's an illusion to be able to think we can get away from petroleum very quickly. But we have to prepare the future. Let's not dream. Let's keep our feet on the ground. We have to keep our feet on the ground by realizing that petroleum will still be used. We cannot just get away with it, but we have to prepare the future. That means trying to develop chemical processes which can use as starting materials plants, biological origins, sources of biological origin, to use them. But this has to be developed and has to be efficient enough. We also, of course, many people and around the world, is the question of having electric cars, for instance, in place of cars which are running with petroleum. Sure, this will happen, and there are more and more electric cars, and it's a big advantage because they are not noisy, they don't pollute, but you need electricity. You need electricity. I insist on that. Electricity doesn't fall down from the sky. Electricity you have to make. You can make it by classical ways which are polluting. You can make it by nuclear power, which is not polluting and which we must use. And India has made great progress in developing nuclear power. For instance, in Chennai, there is a new nuclear plant, which is a very good one. France was very good at that, but they have lost some of it because there was too much opposition. I think that's totally wrong. We need nuclear power. So, as I said, we need to use all the possibilities. We need to use as much as possible solar energy by what people call wind power. It's not very economical, but okay, it can be interesting in certain circumstances. Photovoltaic power to make electricity, that's better, but it's also quite expensive. Nuclear way to make electricity, so we have, and of course, hydroelectric, water, and so on. So we have to use everything available, but we must realize that we are many people on this world. Many people want to make progress towards better life. This involves energy. If you have energy, you can do it. If you don't have energy, you cannot do it. That's number one, and everything depends on that. Of course, you need also the materials, but without the energy, you can do nothing with the materials. I hope to be clear enough.
Thank you, sir.
Yep.
Thank you, sir. Another question from Dr. Srinivasan KP. Can we consider MOF as a supramolecule? Metal organic framework. As a supramolecule.
Organic frameworks?
MOF, metal organic framework.
Metal organic. Yeah, I mean, in some ways they are. I mean, of course, that's the work which has been developed by Suzumu Kitagawa and Omar Yagi, and many people around the world, and Robson in Australia, who was probably the first, did that. And this is, yeah, I mean, the structures themselves are combinations between metals and organics, metal-organic frameworks, as the name says, and then the cavities of these frameworks may be able to pick up other molecules or gases, for instance. And this is, of course, a very interesting field. It's a very important field in chemistry. And it is, you can call it supramolecular. It's also material science. It has molecular
Thank you, sir. Over to Dr. Densley Jones for handling the queries in the YouTube live streaming. Ma'am, please.
Sir, one more question from Shyam Krishnan N. What are the possibilities of existence of different element like silicon-based life form?
This is not a different element. Silicon is an element. There's nothing new. I mean, an element is what I said is in the periodic table. And there are no other elements, as I insisted on. So what is this question? The silicon what?
Silicon-based.
Silicon beads? Sorry, silicon what?
Sir, what are the possibilities of existence of different element like silicon-based life form?
There are no different elements. These are elements which are already existing.
Yes, sir.
It's nothing, means it's not a new element. It's just a new composition of the elements, but it's not a new element.
So another question from Kevin Chaco Muntekel. Can we stop the formation of tobacco mosaic virus?
Can we stop the
formation of tobacco mosaic virus?
Formation of tobacco?
Mosaic virus.
Oh, virus, yeah. Yeah, I think it's a question, I'm pretty sure this can be stopped. As I said, the virus is not living. You must realize that. I think most of you must probably know that. A virus is not living. A virus has to penetrate a cell to use some of the machinery of the cell to reproduce itself. So a virus is a dead object. But of course, it has all these possibilities of interacting through non-living processes of matter. They are non-living, as I said. And so we have to find now ways, either a drug, which will be able to block a given function necessary for the virus. For instance, a virus has enzymes called proteases, and inhibitors of these proteases would be a possibility to make a drug, a pill, which won't stop the virus. And the other obvious one, which is very much now in development, is to find a vaccine, which ultimately hopefully will be found soon. That one finds it, I'm pretty sure, but the question is, how efficient will it be and how long will it take?
Thank you, sir.
Hello? Hello? I don't hear you.
Thank you very much, Professor, for your enduring and patient interaction and clarification of doubts in an obvious manner. Now the interactive session is over. We know that time is scarce, so let us wind up the session. Now over to our head of the department, Dr. J.M. Francis, to propose the vote of thanks. Over to you, ma'am.
Good afternoon, all. We are gathered here for the third lecture of the Nobel Laureate lecture series organized by the Department of Chemistry, Marathinicius College, Kodamagal. It is indeed great to hear from the stalwart Dr. Jean-Marie Le about the steps towards complex matter chemistry. Though complex, he made it easy through his simple and lucid talk as well as his presentation. The session was very informative, and his expertise and ardent love for the subject gave us a new experience of the topic. On behalf of the entire department, as well as the college. I thank you, sir, for the love and support that you have shown to us. We
My
also,
pleasure.
we
Oh,
also,
sorry.
we also look forward to further future collaborations. Next up is our very own Professor, Dr. Vinny Varghese, Secretary, Marathanesius College Association, who is the driving force behind every venture of the college. I thank you, sir, for your incessant support and drive towards excellence. Our principal, Dr. Shanti Evra, cannot be forgotten at this moment. For the constant motivation and cooperation, I thank you, ma'am. Next is the faculty of the department, whose efforts shouldn't go unsaid. Even in unfavorable circumstances, we have stayed together and continued the dissemination of knowledge. Special mention to the coordinators of this lecture series, Dr. Manchu Kurian and Dr. Binu Varghese, who have turned the tables and provided us a productive endeavor. Together with us, as a constant support and help, we have Evin, Abel, Teddy, Bristow. On behalf of the department, I thank you all for your support. Finally, as the lecture ends, I thank all the participants who are present here. The love for the subject and the drive towards new knowledge have brought us together. I hope we continue this relationship to the very end. Once again, thanking each and everyone, I remain.
Okay, thank you very much for this invitation. I enjoyed it. I would like to ask you also one interesting thing is that I noticed that most of you professors are women, ladies. That's very good. That correct?
Yes, sir.
Yes, sir.
Yes, sir. Most.
Actually, we are in a women-only department.
You are only women in the department, chemistry.
Chemistry department.
Yes, I saw that. I looked up your site. Very good. You should continue on, huh? Okay, so thanks again, and I hope that the people who have been listening have been learning something. I will be in touch again with another Chinese in China, but also in India. And in India, I have a next talk next week, another talk in Delhi. So, okay, I wish you
sir.
a day. As you are very far away from me, but I see you, and okay, I would have liked to get there, but it's unfortunately not possible right now. Okay, thank you very much to all.
Thank you, sir. Thank you, sir. It was very informative, as well as very inspiring also.
Good. Bye-bye.
Bye.
ഇത് മാത്രം ഓഫ് ചെയ്താൽ ലീവ് ചെയ്യില്ല.
Bye-bye.
Okay, thank you, sir. Thanks.
An important announcement. We are planning another session of Nobel Lecture Series on October 8, 2020 at 6:30 p.m. Indian Standard Time by Professor Anthony J. Leggett, 2003 Nobel Laureate in Physics from University of Illinois, US. The details will be shared later. Stay tuned and stay safe. Thank you. Once more, thanking you all.
Hello, madam. Excuse me. ഹലോ മാഡം,
ഹലോ.
ഫീഡ്ബാക്ക് ഫോം സബ്മിറ്റ് ചെയ്യാൻ പറ്റുന്നില്ല. സബ്മിഷന്റെ സമയത്ത് അത് അതുപോലെ സ്റ്റക്ക് ആയിട്ട് നിൽക്കുവാണ്.
സബ്മിറ്റ്
ചെയ്യാൻ നോക്കി കുറെ പ്രാവശ്യം, സബ്മിഷൻ ആവുന്നില്ല.
ഓക്കെ, 30 മിനിറ്റ്സ് എന്തായാലും ഒന്നുകൂടെ ഒന്ന് ചെക്ക് ചെയ്യട്ടെ കേട്ടോ.
അത് ഗ്രൂപ്പിലോട്ട് ഇട്ട് തരാൻ പറ്റും അങ്ങനെയാണെങ്കിൽ?
എങ്ങനെ? അതായത് യൂട്യൂബിലുള്ളവരാണോ പറയുന്നത്?
അല്ല, സൂമിൽ തന്നെയാണ് പറയുന്നത്.
സൂമിൽ തന്നെ അല്ലേ?
സബ്മിറ്റ് ചെയ്യാൻ പറ്റുന്നില്ല.
ഇവിടെ നിന്ന് ഇപ്പൊ നോക്കിയിട്ട് സബ്മിറ്റ് ചെയ്യാൻ പറ്റുന്നുണ്ടല്ലോ.
വേറെ 1-2 പേര് അതിനകത്ത് പറയുന്നുണ്ട്.
ഒരു പക്ഷെ ഒന്നിൽ കൂടുതൽ റെസ്പോൺസ് ഒറ്റയടിക്ക് വന്നതുകൊണ്ടായിരിക്കും. ഇവിടെ ഇപ്പൊ നോക്കിയിട്ടും അത് ആക്റ്റീവ് ആണ്, ലിങ്ക് ആക്റ്റീവ് ആണ്.
ലിങ്ക് ഒന്നും കൂടി ഇടോ അതില്?
ഒന്നോടെ പേസ്റ്റ് ചെയ്യാം. മെസ്സേജ് പോസ്റ്റ് ചെയ്തിട്ടുണ്ട്, ഒന്നോടെ ശ്രമിച്ചു നോക്കുവോ?
നോക്കാം.
ഇപ്പൊ കട്ട് ചെയ്യില്ലേ? 10 മിനിറ്റ് 10-15 മിനിറ്റ് കഴിയുമ്പോൾ കട്ട് ചെയ്താൽ മതി. വിളിച്ചു പറയാം കേട്ടോ.
സൈൻ ഇൻ ചെയ്തിട്ടില്ല അല്ലേ? ഒന്ന് സൈൻ ഇൻ ചെയ്തു.
അവസാനം വരെ ഇതിനകത്ത് വൈഫൈ പോയി പെട്ടെന്ന്. ആ സമയത്ത് വൈഫൈ പോയി. വൈഫൈ വേണ്ട ഓപ്ഷൻ കാണിച്ചു. ഇത്ര സമയം പോയി ഒന്നും ചെയ്യാൻ പറ്റുന്നില്ല.
സൈൻ ഇൻ ചെയ്തായിരുന്നോ? സംതിങ്ങിന്റെ അവിടെ കൊണ്ട് ക്രിയേറ്റ് ചാനൽ. ടീച്ചർ എൻ്റെ ഇടയ്ക്ക് മെയിൽ കയറാവോ? ഇവിടെ നോക്കിയിട്ട് ജിമെയിൽ ഒന്ന് ഇവിടെ ജിമെയിൽ ചുമ്മാ മോസിലേക്ക് കയറി കുഴപ്പമില്ല. എന്നിട്ട് ഇവിടെ നോക്കി ആ ജിമെയിൽ ഇൻബോക്സ് എന്നുള്ളത് അല്ല, അത് അതിലൊന്ന് ക്ലിക്ക് ചെയ്ത് ട്രൈ അഗൈൻ സൈൻ അപ്പ്. ഇത് ഡീസൻ്റ് അല്ലേ? അതിൻ്റെ അത് എവിൻ കമ്പോസോട്ട് എവിൻ ബെന്നി.
ഇത് കിട്ടാൻ മാത്രം പോരെ?
എവിൻ ബെന്നി സോറി മാറി റെസിപ്പിയൻസിൽ എവിൻ ബെന്നി കിടക്കണം.
സബ്ജക്റ്റ് ഇല്ല കേട്ടോ, ഇട്ടിട്ടുണ്ടേ. അത് സൂമിൽ നിന്ന് ഞാൻ പോയി.
മാഡം ഈ ഫീഡ്ബാക്ക് ഫോം എവിടുന്നാ കിട്ടുക?
ഹലോ
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 മാഡം ഈ ഫീഡ്ബാക്ക് ഫോം
കേൾക്കാമോ?
എവിടുന്നാ കിട്ടുക?
കേൾക്കാമോ പറയുന്നേ?
ആ കേക്കാം.
ആ ഫീഡ്ബാക്ക് ഫോം ഇപ്പൊ ലിങ്ക് പേസ്റ്റ് ചെയ്തിട്ടുണ്ടല്ലോ സൂമില്.
നമ്മള് ഇനീഷ്യലി തുടങ്ങിയെടുത്ത് പോവാണോ?
അല്ല, ഇപ്പൊ ഒരു 2 മിനിറ്റ് മുന്നേ ഈ സൂമിന്റെ തന്നെ ചാറ്റ് ബോക്സിൽ പേസ്റ്റ് ചെയ്തിട്ടുണ്ടല്ലോ.
ചാറ്റ് ബോക്സിൽ നോക്കട്ടെ.
ആ കിട്ടുന്നുണ്ടോ?